computer. There, it's recording, and then I'll share the screen. Where's the share screen thing? Share screen. There, PowerPoint. Let's start that. Okay, so let me show you the PowerPoint presentation. Oh, there's a problem with this. As you showed me, Paul, online last week, for some reason, when I share my PowerPoint slide, it didn't share properly.
Maybe you still full screen. That's a little green dot.
Okay. That'll do it. And I can see myself, but can I see other people? Well, hopefully it's okay. Okay, so this week is the second lecture of sterilization. I started talking about sterilization and disinfection last week. When I say sterilization and disinfection, there's two levels of getting rid of your contaminants. First level is disinfection. Second level is sterilization. And those are review questions they have coming up. I don't know if I have enough slides for 2 hours. We might. So there's 60 slides. There's usually I have about 70. If the class ends early, oh, that's okay, right? Okay, so food sterilization and disinfection. What did we talk about last week? There's 3 questions, and I have 1, 2, 3, 4, 5, 6 people for questions. Lindsay, Jayun, Elliot, at the back, great. Hamish. Hamish isn't online yet. Aaron here, and Matthew. Matthew's not here. Okay, so I have most people online answering questions, but Elliot, I don't have anybody else. So the other participants, you can see, here's my list. It's me and Robert so far. So, and like I said, I haven't had to answer any more invitations to get into class. So I think it's just you and you out of the list of 1, 2, 3, 4, 5, 6 people that I've been asked questions for. Sorry. That means that everybody in class gets to ask the questions, I guess. I should record names then. That way, when your name comes up later, I'll just take it off the list. Okay, so first question. Now, before last week when I was talking about Maple Leaf Foods, and Maple Leaf Foods had a great big problem in 2008. They had a massive food recall. And the question is, what were the events that led up to that? And I'll let that go to you.
So they had a big listeria outbreak from one. So people became sick, and then they had to voluntarily call that public and pretty much people they were all about sterilization. It makes it hard to sterilize because you have to check it for it to not.
Normally we don't cook deli meats unless you're putting it on pizza or something.
Exactly.
Yeah.
I think it wasn't responsive to sterilizing, which is why it's harder to.
I didn't quite catch that, but yeah, I think you're saying the right thing, and it's hard to get rid of.
Yeah. So. Food, it was food
Listeria is something that causes an infection inside. Yeah, so it's not known for its toxins. It's known for its infection. So if you get a listeria infection, you become weaker to the point where it can be fatal, and especially in immune compromised people, babies, adult, seniors. Thank you. Oh, Robert is online. We can see that you're there, Robert. Neil's back. Thanks. Much boo. Chat. Maybe they have questions. Hello. Robert says hello. We have 11 people in class, and we're just waiting for more people online. And Elliot just answered the first question about Listeria monocytogenes being the infectious agent at Maple Leaf Foods. And second question I got here, what are the symptoms of Clostridium botulinum? Go ahead.
Is this one with descending paralysis?
Yes. So the symptom is descending paralysis. You start from droopiness in the eyes to loss of muscle tone. Yeah, and it's very dangerous.
Vision, double vision.
You asked me that last time. I don't know about lockjaw and botulinum. I've seen images of lockjaw, the tetanus disease, and how people just become contorted from that bacteria. Israel or Will, if one of you two guys could just close the door, just to quiet it down out there. What kind of foods do we commonly associate with this bacterium? Pardon?
Honey?
Honey is one. Seafood, canned foods. And canned foods is honey and canned foods are really important with this because canned foods, you would expect them to be sterile when you get them home, but it may be the case that the bacteria is still active inside. If you have a bulging can, that's the reason to get rid of it. Dented cans like you were talking about, because you could be taking a chance, but
No.
is there a discount on dented
I don't know
cans?
if they do it anymore. They used to. A lot of places will do them because people will not buy them.
Oh, okay. So
I will. I'll throw them away.
Oh, there's someone reading.
all right, Hamish. Welcome, Hamish. And next part of this question, is this food infection, food intoxication, or food poisoning? Yes, it's based upon a toxin, one of the most deadly toxins out there, right? They say that if you had 5 grams of this, you could kill everybody on Earth. So it's a deadly toxin. I don't know if that's true. I saw it on a TV show. Resident Alien. I don't know if anybody's ever heard of that one.
I do think it is actually true because I've heard that before from more sciencey, actually, like documentary type shows.
Yeah.
Okay, it's not Facebook official.
And Hamish, since you're here right now, let's see if you can answer. What were the events leading up to the water treatment failure in Walkerton? Because I've got you as answering questions. So I'll say, Hamish, can you answer this question 3? Yes,
Sorry.
I'll
From africancheck.org, it says a tiny amount, perhaps as little as 4 grams of the toxic botulinum can kill all of the billions of people.
So it's an estimate.
Let's not try it.
I've only got 3 grams.
Keep it to yourself.
Okay, Hamish says he's not sure. Okay, I'll save another question for you. Somebody else want to lead? Go ahead.
So the Walkerton crisis was when a really large amount of E. coli as well as Campylobacter jejuni entered the water supply
That's right.
in Walkerton, and that was a result of like days of heavy rainfall. So that surface runoff entered the water system, which also overwhelmed the system. So I guess
From the manure, from the.
with Walkerton being primarily rural. So that's what was Washington, and basically like the municipality just had really inadequate water testing practices, and also they have also like dishonest practices where they would like
Say
lie.
they tested, but they
Yeah.
didn't test.
Exactly.
Yeah.
So
Kind of
that's what
important.
many people got sick. I think like under 10 people died.
And from my understanding, some of the people who got sick in Walkerton are still suffering with the infection. Like it, so that's 20 years ago. They're still not quite normal. So it was a severe intoxication, I guess. Next question. The next part of the question: what characteristic of the organism makes it harmful?
So I think E. coli in particular, it can survive cold environments, and it can also be spread person to person, rather than just a person being in contact with food or water.
Not so much person to person. I'm looking more. Do you remember what the protein is called for E. coli? It has a special kind of name. No? Okay. Do you remember what, is it food intoxication or food infection?
I've got it written down and I copied it off your slideshow.
It can be an infection, but it's also more the really harmful part are the toxins. So the Shiga toxin is the toxin that I'm thinking about. So this is a toxin that is found through a lot of different bacteria. And the Shiga toxin, if you take it in, it can cause havoc in your intestines, causing bloody diarrhea, like that sort of nasty kind of condition. So it's food intoxication. This is one of the main way. Oh, what was the name of the? Oh, you told me already. It was Campylobacter jejuni and the other one,
E. coli.
E. coli. And there's one strain in particular. It's the 0157H7 strain that they talk a lot about. So if you see that in the news, it's because it's one that's contaminated feed coming from. It's infecting a lot of animals. Hey, there's another person coming. Matthew. Okay. Thanks, Aaron. Next question. What is sterilization? And so I saw Hamish, you're online. And Matthew's online. 2 questions for you guys.
Which question do you want me to answer?
Number 4. What is sterilization?
Sterilization is the entire elimination of any living organisms off of a given surface or object.
Good. How is that different from disinfection?
I assume that disinfection would imply that it's been infected with something and that like you'd be killing like a pathogen, like something that's like only harmful.
Close. So sterilization is you're getting rid of everything. Disinfection, it's like an amount. You're removing some of them, not all of them. Okay. Thanks. That's Matthew. Hamish, last question for you. What's the difference between cidal and static? Like something that's fungicidal or fungistatic? It's kind of the same as sterilization and disinfection.
Um, I'm not even sure.
Yeah.
Sorry.
Okay, well, we'll come up to questions a little later.
Thank you.
No problem. And the difference is cidal's like killing. So it's more like sterilization. And static is you can kill some of them, but some organisms may still survive. So it's just a matter of killing. So let's take a look at next slide. Okay, so today I'm talking about 5 different things for sterilization and disinfection. The first thing being treatment with heat, hot and cold treatment to food to get rid of organisms. So, or reduce the contamination level. So you could do that by high temperature, also by freezing. If you're freezing stuff, you're reducing the ability for the organism to multiply. Next part of the lecture is radiation. Then I'm talking about filter treatments, chemical treatments, and then finally antibiotics. So the first part is the hot and cold treatment of food. And the first picture I've got here is a picture of an autoclave. So this person on the left in the image, you can see the autoclave, he's pushing a cart of media or food, could be jam, into an autoclave, and he's pressure cooking it. And like I was saying earlier, it's really important to remove the threat of Clostridium botulinum, especially in canned goods. So you can see that this is a good way to get rid of that. Autoclaves sterilize liquids, they sterilize glassware, they sterilize media for growing microbes, they also sterilize food, soil. So autoclaves sterilize a lot of different objects and items. So you're using heat and steam to kill off everything. Here's the steps for autoclaving something. So first of all, you get it to a really high temperature, say 120 degrees, and 121 degrees is always kind of the reported temperature. I think because 250 is the Fahrenheit temperature equivalent. So 250 Fahrenheit or 121 Celsius. That's why it's so precise in Celsius. You know how you have food items at home, you buy a can of Coke and it's not 400 milliliters, it's 355 or 354 milliliters. So it's like a precise number as opposed to a rounded number. I think that's kind of the same reason we typically talk about sterilization with the temperature of 121 degrees Celsius is because they're rounding or they're using 250 Fahrenheit. The next is the pressure in kilopascals. It's 105 in imperial units. It's 115 psi's. So it's under a lot of pressure, and you put it under a lot of pressure for 20 to 40 minutes.
115 or 50?
Pardon?
15 psi or
15 psi. I think that converts. I did the conversion. I think that's
I thought you said 150.
105 kilopascals, 15 psis. So what you do is you put everything into the autoclave, kind of distribute it evenly, and then you seal the chamber. And there's old autoclaves out there where it's just operated by valves and thermometers. And what you do is you seal the door, allow steam to go enter the chamber, and then you just kind of stand there and wait until you see the temperature gets up to 121 degrees. Because what you're doing is you're capturing steam going into the chamber. And it also, you have to watch the pressure, make sure it goes up to the right level, up to 15 PSIs. And if you do autoclaving, if you do use a pressure canner at home, you know, the instructions are usually get the pressure canner to 11 pounds per square inch or more. There's like a little gauge on some pressure canners that allow you to get it up to the right pressure. And you cook that for 20 to 40 minutes. This is for liquids. Dry items have a different time. And depending on how thick the media is, you may have to go through a longer cycle. This is all done by experiment to figure it out. Then what you do is after you've sealed off the steam and it's allowed the time to elapse, you allow the pressure to release slowly. And the reason you're letting the pressure out slowly is the material, the liquids in there are under pressure. So they're at a boiling temperature, but they're not boiling because they're under so much pressure. So it's not bubbling rapidly inside, but they're at a higher than normal bubbling temperature. And what you do is you release the steam slowly and you allow the pressure to come down and the liquids will be still hot and you kind of try and match the bubbling with the release. And this is what the automatic autoclaves do is you just press a button, walk away, and it gets up there, allows it to stand at that high temperature and comes down at a very smooth level. You can't use an autoclave on something that's plastic or paper. And the reason for autoclaving not working with the plastic is you could melt it. You have to have something. I guess you can have plastics that autoclave, but they have to be heat tolerant plastics, right? So if, some research labs will have a display where they show the melted things that students have put into the autoclave to show like, please do not put these items into the autoclave because if you put them in, cook them for 20 minutes at this high intense heat, the plastics will come out melted. So do not put plastics into an autoclave. So heat is a common sort of treatment for foods. And I don't know if you guys do home canning, open water, hot water bath, or pressure canning, but this is very, very common sort of way of preparing your food that it's going to store for a while. But if you cook food, you're not sterilizing the food. For example, if you're grilling steak or boiling food, microwaving it, roasting it, you're not sterilizing the food. What you're doing is you're just cooking the food. Sterilizing is a different idea where you have to get it really hot for a long period of time, kill off all the bacteria, then reduce the heat, okay? Cooking reduces the number of disease-causing microorganisms, so it does help. But when you heat sterilize, it's removing the bacteria totally. So if you boil foods that kills the vegetative state of microbes, but it doesn't kill off spores. Typically, spores can still survive. So if you're thinking about open water bath canning, where you have a big pot of water on the stove and you put your cans of jam inside, what that'll do is it will cook it. And if there's any, like I said, vegetative bacteria, it'll kill those off, okay? If you put it under pressure, like in a pressure canner or an autoclave, that'll kill off the vegetative bacteria and spores. So it's both parts of the bacteria that you're killing off. And that's why it's good for preventing botulinum, because botulism has spores. And so, or is it botulism? Yeah, I think so. Pressure cooking is also maybe I have to check that. Does botulism have spores? I said it, and I'm thinking, why am I saying that? I'm thinking maybe anthrax has spores. Somebody want to check that for me? Clostridium, does it have spores?
Yes, they make spores.
Okay, thank you. Yes, they do make spores. When you're pressure cooking food, it's kind of like autoclaving, and that's making your food sterile. So if you put your, I don't know if you're making stock, vegetable stock, turkey stock, beef stock, put it into the autoclave, or tomato sauce, put it into your pressure canner. It's a good way to remove all of the vegetative bacteria as well as the spores. And it says here, home foods are difficult to sterilize with home equipment. And that's because a lot of people don't wait the entire time. They're kind of impatient. We just want to get through to the next step. So I guess the idea there is to follow the directions as close as possible. Okay, so here's a heat treatment explanation. What can affect your sterilization and disinfection? It can be the number of microbes. If you have lots of microbes there, that's harder to get rid of them. If you have difficult to remove microbes, that's also something to think about. Time of exposure. If you don't leave the exposure long enough, you'll get these bacteria being a problem there. And environmental factors are also important, like pH, temperature as well. And if you can see here, there's 3 graphs. What does it say there? Salt of, I picked a really bad graph. Can I see that? Start of cooling phase. So they're all going up to 110 degrees Celsius. And then first one, after 60 minutes, they're letting it cool.
So it took 60 minutes for it to reach the 110, then B took 90 minutes, and C took, what was that, 125?
Right. So they're taking different amounts of time. Core temperature.
Goes spots in the plants. So that's when determine whether it's pasteurized or. So it's the amount of time it takes the cool spots to get to the desired pasteurization temperature and held there for some time.
Right. So if you heard online, Tavechko was saying the temperature in the cold spots of the can needs to reach the higher temperatures. Oop. Get this back up. And you have to heat the cold spots within the can. The cold spots are the bad parts. Next slide. Pressure canning is like autoclaving. I just said this. I think I've home foods are notoriously difficult to sterilize with home equipment. So you have to read the directions, follow the directions, and don't make up the directions just because grandma did it this way. Try and follow a protocol that is a safe protocol. I also included just a slide there, sterilization. I don't know if you can see that. This is the removal of TSE. TSE was the transmissible spongiform encephalopathy. That's the prions, like Creutzfeldt-Jakob, mad cow disease prions. How do you get rid of them? It's very difficult. They're finding that if you add a lot of heat, it doesn't really get rid of them. So if you add sodium hydroxide, it does. And that's what this slide does. And somebody asked me that question once, so I found that and put that up. Yeah, go ahead.
No, if it's more difficult to get food sterilized by cooking them at home. By the way, people get less sick by eating food prepared food than eating industrial. Well, if it's sterilized, it's sterilized.
Maybe, I don't know. Maybe it's time.
Tell you what, don't eat. Eat food prepared food so that you
Good question. The question is, why is it less people get sick at home eating their own cooking than people get sick from industrially prepared foods? And this could be time, like, so the amount of time that something that's prepared from an industry that gets to your house is longer. So maybe the bacteria there might have multiplied. Whereas if you're having it at home, you've cooked it, you've made it the next day or something. So like,
And the amount too, if you have more of it, it's more likely to be contaminated.
You think of like a deli meat goes from whatever abattoir through legally, then to the grocery store, then it goes on to that dirty slicer.
So many touches, and so many touching points along the way for.
So it's your vectors.
yeah. Where is it picking up these bad bacteria?
So there's so many potential points of contamination. That slicer's gross.
Is that your freestyle? Like, what's
I think we're
those,
supposed
how
to.
do they get?
Yeah.
They're supposed to.
Teenager does it after hours.
I was gonna say supposed to be cleaned and actually properly cleaned are two different things.
Yes.
It's a matter of people doing what's required, doing their jobs, right? Okay, so here I have an explanation of high HTST treated milk and UHT treated milk. So HTST, high temperature, short time treated milk. This is a pasteurization. This is what we commonly refer to as pasteurization. So what you want to do is you want to pasteurize milk. That way it kills all of the vegetative bacteria inside of the milk. You heat the milk up to 72 degrees Celsius, 15 seconds. That's all it takes. That's pasteurized. And then you let it cool down. If you do an ultra high temperature treatment, what you've done is you've taken the milk and it doesn't go up to just 72 degrees. It goes up to 140 degrees Celsius. So this ultra high temperature is under a lot more pressure, really under controlled conditions, but it's only cooked for 4 seconds. So you get it up to this temperature, 4 seconds, and you allow it to cool. If you treat your milk with a UHT treatment, you don't have to refrigerate it. I don't know if you've ever seen Tetra Packs on grocery shelves with milk in it. You can buy it like that. And this has been ultra high treatment.
And the packaging as well. Wendy was very clear about the aseptic packaging and the fact that they use aseptic packaging and would also contribute to the long shelf life, because you're starting with a clean vessel.
Great. So you're starting with the clean packaging, and the clean packaging helps as well in that case.
There's no possibility of transferring the bacteria with the aseptic packaging.
UHT is called flash pasteurization in the industry.
Pardon?
UHT is called flash pasteurization.
Yes, yeah, flash
in the industry.
pasteurization. That's right. I have a couple of YouTube videos. If you're interested, I have 2 from Canada, 1 from New Zealand. If we have time at the end, I'll share them with you guys. But they just kind of go through a plant, talk about how it comes to the plant in the trucks, how they remove it into the pasteurization chambers, and then they take it, bottle it, sell it, bottle it. It doesn't happen very much anymore, does it? Bottling, plastic bag or cardboard container it.
Bottling actually happens a lot better.
Bottling, you can buy bottled
You
milk.
can buy bottled milk here, but it.
I know.
I think it's Ontario and BC that only have bags left. There's bags are rare. They're not very common.
Really?
Really, most places have jugs or cardboard boxes.
Yes.
You definitely will not
Bags
find
is
bags
like
in the
an
States.
Ontario and Quebec thing.
I was gonna say Ontario and somewhere else, but I
Yeah,
couldn't remember
Quebec.
where.
I grew up in Alberta. We had bags.
They don't now.
Yeah, they changed to plastic containers.
Yeah, I was gonna say everywhere seems to contain to be plastic containers because they're
I thought it
easier.
was all across Canada.
So did I. That's where
Yeah,
I
I
learned
thought
that.
it was a Canadian thing, because
Yeah, no,
but it's not. It's like Ontario and Quebec thing.
For plastic bags still.
The plastic bag.
Yeah.
Yeah.
Some company just clinging to it, right? That's-
It's unusual.
Don't
I
get rid
guess,
of the bags.
for
you give somebody a bag of milk that's never seen a bag of milk in their life. and
They
tell them
don't
to
know
open
what
it,
to do with a plastic bag of milk.
And they
Yeah.
get lost, really lost.
So we're talking about plastic bags. If you are curious online what we're talking about, because plastic bags are used for storage of milk.
Yes.
I can see the point of using plastic bags, far less plastic than in plastic containers. Because you're using the harder plastic, more of it. So it's got to be stronger plastic.
Harder plastics is recyclable.
That's true. Cooling and refrigeration, 2 other ways of treating food with temperature that we can use to slow down bacterial contamination. Refrigeration slows down the growth of your microbes. And this has only been around, if you can think of this, for about 150 years. There may be less. There haven't been any refrigerators before the 1900s. This is new technology. Pardon?
It was in the 18th of
18 something that they invented refrigeration. Yeah.
in the Germany, in the process. in Germany.
Yeah.
You know, that was sponsored by Heineken.
Oh, yeah.
Yes.
So Heineken was really important for refrigerating beer, really important.
Looking for how to preserve their beer. They used to use snow from Norway to
Keep it
preserve
cool.
beer. So they were looking for how to So they sponsored the University professors in Germany. So he came up with the refrigeration process.
Cool. I didn't know that.
The
So a
first time
refrigeration
he
process is like Tobechko was saying,
Yeah.
invented in Germany for Heineken. Cool. And like I said, this hasn't been around for very long. 100, I don't know, 20, 100 some years. I know that Einstein had a patent for refrigeration. I don't know if it was like an air conditioning or, but he was involved in one of these patents too. With the refrigeration, now we can have at home fresh fruit, fresh salad, fresh milk, dairy products, or anything that's susceptible to being warm for too long. Mayonnaise, we can have all of these things stored at home. A lot of food poisoning bacteria live and grow between 0 to 54 degrees Celsius. But their really fast growth is over 27 degrees Celsius. So if you keep foods cooler, they don't grow as fast. It's important for foods also to cool quickly. And like I have here, it prevents outgrowth of Clostridium. Freezing is another way that we use to preserve foods. We preserve lots of food with freezing, right? We have vegetables, meats, whatever's in your freezer right now. Is just think about it. We preserve our foods this way. It's used for preserving lots of different kinds of foods. And it's oftentimes used for preparing or freezing prepared foods. So if you think it's not for unprepared foods, typically, it's for prepared food. So if you've sliced up your French fried potatoes or your celery for soup stocks, or you've got some sort of prepared vegetables, you could freeze that. But typically you don't have celery stalks, whole peppers. You don't typically freeze the whole vegetables. You typically freeze prepared stuff. And I have an example here. French fries and hash browns are stored in the freezer, but you don't store potatoes in the freezer. Because potatoes, you can keep them on the shelf in a cool place for months. So you can have things in unprepared things in large volume on the shelf. Okay, so here's 4 questions. And we have Matthew's online, as well as Hamish. What's the difference between a hot water bath canning and pressure canning? Well, Hamish, you haven't answered the question yet, so if you could, are you there?
Yeah, the hot water canning, it just uses hot water to sterilize. And the pressure canning uses like pressure and hot air to sterilize. Or yeah.
All right, so you're using a sealed container with your pressure canning. Which one kills more organisms?
The pressure canning.
Great, good, okay. I'll leave this, Matthew or Aaron, or Elliot, if you want. Question 2, explain how an autoclave works. Thanks, Hamish. Somebody want to just go over the steps of making an autoclave work? Oh, yeah, yeah.
You seal the water temperature. You fill it with steam, and then the steam will reach the time. Move that to my time. And then the pressure passes off.
Good. And I've gotten Elliot to give me these steps. Thank you. Describe 2 methods for pasteurizing milk. So that's the high temperature, short time, and ultra high temperature. So I'll leave that to Matthew or Aaron.
Sure, I can talk to HTSC. Basically, it's seen a lot with like traditional pasteurization of milk. So it's typically at 72 Celsius for 15 seconds. What it does, it just lowers the microbial numbers, but not necessarily killing all of it. But at least it prevents food from spoiling. Ultra high temperature, it's at a higher temperature, but for a shorter time, and ultimately sterilizes.
Great. Do you remember the temperature?
140.
Yep. 140 degrees for just a few seconds, 3 or 4 seconds, I think. Okay, yeah. Everybody in class, why do we freeze French fries but not potatoes?
French fries are prepared.
And Cass, you said,
They're cut. They don't have their skin on them anymore.
yeah. Thanks. So that's that slide. Okay, next section, radiation. So I've talked about high temperature use for sterilization. Next part is going to be radiation. Next part after that's, like I said, filter, pressure, treatment, then chemical treatments. Okay, so irradiation. Irradiation is exposing your food to an ionizing radiation. And I've never seen this being done, but I've heard of it being done. This is used to preserve food. Has anybody here been in a plant where they've done this? Yes? Go ahead. Yeah,
It's like you have what you call it, not to say but the chamber, I can't remember what that chamber is called.
an
Then you have-
ionization chamber, maybe.
Yes. And so chamber, then you have the source of the ionization focus, like it's used to kill there is what you call there is this what I used to make there. You have to sterilize it first.
Right.
So that is bacteria free. Before you now dilute your beer, dilute the beer that has been fermented. So that water, since the water cannot be heated with any process anymore, it's cold, it's very cold. So they use this to make it bacteria free.
Huh.
Pass it in the chamber. Yes, it's very cool since you have to, when you're filtering, the beer has to be cold. And the water cannot be heated to like get the bacteria. So you have to use this process to keep the water cold and still,
Yeah, so
remove all the microbes.
not everything can be heated, and beer you can't heat, like Toby's saying. Yeah. So, and if you want it cold, you have to find a way to sterilize it so it's cold. So one way of sterilizing is irradiation, and the other way I've got it a little later is filtering.
So chamber, as the water passes through, the chamber has the source of
Irradiation.
Made like glass. So it's so as the water passes through, it's sterilizes as it goes.
Cool. Yeah. Natalie's got it.
Am I right to assume irradiation would kill spores, because I know it's used in a lot of commercial putting it out on the market. So I'm wondering if that's for spores.
Maybe. I do have it here. I don't remember. Okay, let's take a look.
For like medical stuff too, like your Band-Aid, brand
Yeah.
Band-Aids. So it's not like they disinfect each individual one, they take pallets of them into a radiation chamber and then blast them with gamma rays.
Right.
That's how they're stored.
And this is a question that's coming up. What kind of radiation do they use? Gamma radiation. Good, yeah. Okay, so radiation is emitted by a radioactive source. The food does not become radioactive. So you have the gamma rays going through the food, but what's radioactive is the source. It's not the food. So the beams of gamma radiation go through the food or the water, in Toby's case with the beer. And Natalie, what were you saying? Okay, sorry.
Where
Yeah.
am I saying? How long ago was that?
1 minute. Sorry, me too.
I'm on to something else in my head.
Okay.
Irradiation.
We take a break.
Are you sure you're not wrong?
I have to PhD, I'm on to something else. If it kills spores.
Oh, right.
Because I know they irradiate the cannabis, and I'm assuming that's because of any powdery mildew or what was it we had last year? Really bad,
Vegetative,
bronchitis and stuff.
vegetative, when I say vegetative, I mean the actively growing stuff, they're killing all of that off. But I don't know what spores, we'll see. Irradiation is used for non-food appliances like medical devices, like Paul was saying, you put them into a great big chamber. If your medical devices can stand up to other chemical treatments, they'll use that too. Next slide. Here's a symbol for something that's been irradiated. It is a circle with what looks like to be a plant inside. This reduced pathogens in foods, depends on the dose. Some or all microorganisms, bacteria, viruses can be destroyed, slowed down, or rendered incapable of reproduction. So they might be damaged to some degree, and so they can't spread. Reduction of pathogens in food. Some foods are irradiated at doses to ensure the spoilage is sterilized. Okay, where's the thing about spores? Irradiation prolongs shelf life of foods. It delays your ripening of fruit, sprouting of vegetables, slows down ripening, slows down enzymatic production in food as well, so it doesn't degrade enzymatically. Irradiation doesn't change it though. So if something is slowly ripening, it doesn't reverse the course. So if it's fresh, ripe food, it's not going to bring it back to a more fresh state. It's not going to reverse aging. And it says here, it might not destroy toxins or repair texture, taste of food. Typically, when food is being irradiated, pallets of food are exposed to a source. And this is similar to what you were saying. In case of medical equipment, pallets are put into a great big room. So you put a dosimeter into the middle of the pallet someplace, and you pull everything out and check the dosimeters, make sure the dosimeters receive the proper dose. Sometimes radioisotope is lowered into a water-filled storage pool, which absorbs radiation but does not become radioactive. So there's radiation there. Most irradiated food is processed by the radiation called gamma radiation, which is what you're saying. Special precautions are used because gamma rays are constantly emitted. So just because you have the source there and you see the container, doesn't mean that it's, when you take everything out, that the gamma ray is done. It's still emitting the gamma rays, so you have to be careful with how things are moved around the source. Gamma radiation is penetrating, very penetrating, so it goes through everything. And the sources that I've got written down include cobalt and cesium. So cobalt 60, cesium 137. Cesium 137 is used in hospitals. They use it to treat blood transfusion and prevent grafting of diseases. Graft versus host diseases. Okay, so here's a trick question. I don't have the thing about spores. Sorry. Spores and irradiation. If somebody's, if one of you guys want to look it up, see if irradiation kills spores. It does. Yeah, okay. How radioactive are foods that have been irradiated? Okay, trick question, yeah. How does irradiation source ionize microbes? I guess I'm asking, what is the radiation type? Yeah, it's referred to as gamma radiation.
That's what does it do to the microbes?
The gamma radiation will, I think, break down any cellular bonds.
I know it messes with DNA.
And
Sterilization messes with DNA so bad.
yeah, if you break DNA blocks, then it prevents any organism from.
Not fast. Not fast. Not fast. Not fast. I'm thinking, I don't know. It's to immobilize the bacteria.
So yeah, the bacteria are no longer living once it passes the source. So they're alive on one side, they go through the source, and then on the other side, they're dead or dying. Quickly, yeah. Can irradiation remove microbial toxins? If food has toxins already, no, it's not going to remove those toxins, but it can break them down so the chemical bonds are broken. Does it freshen up old food? And the answer is no. Two common radioactive sources for irradiating food include cobalt and cesium. Cesium. Okay, so that's the radiation part, filtration and pressure. I see Robert's stepped out, come back in. So for high pressure, what you're doing in the use of high pressure is you're allowing food to go through a filter of some sort. And you're doing this under a lot of pressure, and the filter is taking out the microbes. And the reason for doing this is with foods that are temperature sensitive. So you don't want to treat any temperature sensitive food to high temperature. You'll cook the food and you won't have a good food product. So it's for allowing cold foods to go through a treatment and then it's sterilized. So I have here pressed inside of a vessel. Exerting 70,000 pounds per square inch, 480 megapascals. Food is processed so it retains its fresh appearance, flavor, texture, nutrients, and at the same time disables harmful microorganisms. So this can be used for orange juice, guacamole, deli meats. Pardon?
Something that's gonna take being squished.
Yep. Yeah. Deli meats. I didn't know about deli meats being squished, but
Mutilate them and smush them into a tube.
yep. And guacamole being squished. That's unusual to me. I can see fruit juice. Okay, so membrane filtration is a technique that uses a porous filter and separates your particles into a fluid. And you have special filters of different pore sizes, and the smaller the pore, the more likely it is to capture bacteria or fungi. And then there's a lot of different kinds of membrane filtration. You can have reverse osmosis, nanofiltration, which is like a really ultrafine sort of filter, ultrafiltration, microfiltration. So it's all just in difference of pore size, microfiltration being bigger than the nanofiltration. When you do this sort of thing, you'd use fluids that could be damaged by heat. So if you don't want to damage your food product, you let it go through a filter. Foods like wine, beer, they get filtered. Have you guys made wine? Did you have to filter it? No, yes?
No, we didn't filter it. We definitely did not filter it.
Well, they can't do it.
We
Decanted it, all of the stuff settled to the bottom. Yeah.
We didn't actually filter it.
We use like the siphon to like just kind of let the sediment go to the bottom and then like pull the wine from the top, but we didn't like properly filter it.
No, they're still sending.
There's still sediment at the bottom. Yeah, yeah, that's fine. Decanting works. Thanks, Matthew. This is good for wine and beer, like I was saying. And if you have a pore size of 0.2 microns, that's going to remove your bacteria. So it's got to be fairly small. When you do this sort of work, you don't just put it through the smallest filter initially. It's usually a stage of big filter to catch all the big stuff, medium-sized filter, and then your ultrafine filter. It's like a couple of steps. You can even get rid of viruses this way too from your material. So in that case, it's done with the nanometer porous filter. And in this case, if you do let something go through a filter that is a nanometer filter, it's really slow. It takes a lot of time. So you have to kind of build that into the treatment of the material.
Apply pressure with pump. With pump.
You could push it through with a pump. Yeah.
That's how, yeah, it's really that you apply pressure with pump. If you want to test soil, if you know quesago, the quesago is like soil. It's gotten from the soil. It's used to filter beer. But I heard it's carcinogenic, so like they are trying to stop it.
Ah.
It's the main filter for beer. Quesago is, you apply, you build it up. As the beer, it traps yeast.
And cleans your beer up, gives you a nice product at the end.
But it's, I heard it's carcinogenic, so most brewers are now trying to change to membrane filter. But the membrane filter is very costly and
Yes.
it's difficult to maintain. So like quesago is like so you pump it, it forms a linear bed, then you filter. If it, after some days it gets blocked, and you wash it every pump.
Huh.
It's so easy to form.
So everybody here's made wine. Have you guys all made beer too?
Not yet.
Not yet? That's coming up.
We are going to take it.
In this slide, I've got a bunch of different kinds of organisms, sizes, and what kind of filtration is going on here. As you can see, it goes from particulate filtration, which is like the really big stuff, and then your microfiltration, small stuff. Micro indicates that it's a micron size filter. Ultrafilter is molecular range, and then nanofiltration, which is like the viral range. And then reverse osmosis, which is almost a chemical range. So you're just removing chemicals. Okay, so, and why do we do this? It's the avoiding of the heat treatment. Not everything can be heated. Some examples: fruit juice, apples, carrots, cheeses, ricotta cheese, butter, ice cream, fermented milk. There's even fermented milk. You can buy this in the grocery store. They say it's finely filtered. This kind of milk is really kind of interesting to me because you can buy 2 liters of this stuff and it'll last for months in your fridge. It's kind of like the UHT milk that lasts on the shelf. Microfiltered stuff can last for a long time. I know one woman who she was a researcher. She used milk in her research, and she'd buy like a 2-liter container of milk every 6 months or so. It would just kind of sit in the fridge. They'd use it up slowly, but the microfiltered stuff can last a long time. And it's also used with beer and wine. Now, you don't want to heat your wine. You don't want to heat your beer. So this is why they'd use filtration. It increases shelf life and reduces production steps. So you can get more food when you're filtering your stuff. There's also low energy costs. So common foods that undergo filtration treatment. What are some? And I'll leave this for anybody. Anybody want to answer? Wine, beer, orange juice. Find it interesting, guacamole. The food that can undergo. That's what it said.
A membrane?
Oh, I don't know about membrane.
No, that was the pressure one.
A high pressure, right, right.
I was gonna say, I would not put guacamole through a membrane.
Baby food? I'm guessing.
Parts of the baby food, maybe.
Some
Strain baby food. That's-
And they, yeah,
soup stocks.
Common drinks undergo membrane filtration. We said beer.
yeah, we covered the
Wine.
drinks, really not food.
What does pressure treatment do to microbes to ensure food safety?
I think it damages the cells.
Yeah, they burst. Yeah. Is there a benefit to using filters or pressure compared with other sterilization methods? No heat. Good. Yeah.
It kills the microbes. It's more spores. It removes the microbes.
Removes the microbes. Yes. Might be less costly.
Less costly, and it takes them. This one kills them. This one filters the most.
Great. Sloughs the bad stuff out.
So heat, filter actually removes it, pressure, heat, kill it.
Okay, two more topics. Let's see, 1224. So we have D&E, chemical treatments. There's a bunch of chemical treatments here. There's gas treatments, liquid treatments, salt treatments, nitrates, acids, sulfur, and then I talk about antibiotics briefly. So chemical treatments. Some chemicals are used for sterilization as well as disinfection. If you can't use heat, you can use chemicals. Problem with chemicals, it can be very corrosive to materials. So you have to kind of judge whether or not you're using chemicals on your food products. It says, ensure the article to be sterilized is chemically compatible, which I just kind of was saying. It can destroy lots of pathogens by using a chemical treatment. Things that make things good sterilants are harmful to humans. You think of something that's a good sterilant, bleach. Bleach is a sterilant. Well, it's a disinfectant. That can be very harmful. Peroxy acid. So here, ozone. Ozone's a very harmful sort of gas. Anyway, so what are some gases? You can use gas to sterilize things. There are reactive gases, something like ethylene oxide, hydrogen peroxide, or ozone. You can sterilize your, I know there's autoclaves that use hydrogen peroxide for dentists. What they do is they clean their tools because the tools need to be cleaned with hydrogen peroxide. And if you think peroxide is a very aggressive oxidizing agent, you take hydrogen peroxide, put it on a wound, it bubbles, it's reactive, like very reactive. Even at a weak store diluted amount, it can be very reactive. So we think that is a good way for sterilizing your medical tools. I have a ethylene oxide YouTube video here. Some of these YouTube videos are almost like advertisements. I'm not saying watch them for the product company. Just here's a product that uses this kind of treatment. So you can see their Medline, a company, is using ethylene oxide to sterilize 16,000 packs a day. So some medical equipment is being sterilized with ethylene oxide. Laminar air is used by microbiology labs. If you've noticed in the back of the microbiology lab, there's this great big metal cabinet with a hood on it. What the cabinet does is it creates what's referred to as laminar air. And laminar air is air that's moving in a sheet-wise kind of way. And what you're doing is you're forcing like a curtain of air down, and you're recycling the air through a HEPA filter. So when you turn the microbiological cabinet on, you sterilize the entire area, and then you turn the HEPA, like the airflow on. And if you put your hands in, your hands are the only thing that are non-sterile. So you can work with sterilizing media, sterilizing plates, all kinds of research is done in laminar airflow hoods. Yes.
I watched a guy and he only grows mushrooms.
They use the fume hoods, laminar flow hoods for treating media.
Yeah, he has a bunch of different filters set up.
Yeah, so it's just a clean way of treating media. You don't want that to get out. Yeah, you don't want your product contaminated for sure. You want clean product. You don't want mold or, yeah. Yeah. Very common in molecular biology. We don't talk about mushrooms very much in this course. It's almost like something that's a lot of students want to do. I know I talked to one student from last year. He's got he's got a food product. He's talking about making a food product from mushrooms. It's like beef jerky, only it's mushroom jerky. I'm not sure. Oh yeah, cool.
It would not texturally be
This is a guy who's 1 year ahead of you guys in the food program. He's shared that with me.
Yeah.
So if you like mushrooms and you're thinking food, that could be something as a project for you guys for next year. So you can work with mushrooms at some point. Okay, next point. Inside of these laminar flow hoods, you could also have flame. Flame kind of disrupts the laminar flow, so you have to kind of keep the flame back away from the opening. Also, you could have a UV light to sterilize the bench, and the UV bulb is just right there at the top. You turn it on, and that sterilizes the cabinet as well. Ozone is another gas that's used to disinfect surfaces. And with ozone is a very oxidizing gas. It's very aggressive. It's a toxic gas. And if you think this is a gas that's more oxygen than oxygen, so it can be very reactive. You want to be careful with flame, with ozone. The advantage of using ozone, it's destroys a lot of things, even prions. And you don't have to handle dangerous chemicals because if you use ozone, you've sterilized it and it just reduces down to oxygen. Reactive ozone means that it's destroyed right away with if you use a catalyst. You have to run it over a catalyst and it goes back to oxygen. So it doesn't last very long. I know it was even used in water treatment plants. Yeah.
That's where I was thinking.
I'm thinking food wise, I'm not too sure, but if you'd want sterile water. So you could boil water, but you're not necessarily getting everything. Autoclave water, it's using a lot of energy. Ozonating water, you're just bubbling ozone through it. As long as you use a catalyst to get rid of or reduce it. Chemical treatments for liquids, sterilants include hydrogen peroxide as well as peracetic acid. I talked about peracetic acid before. Just briefly, it's one of the acids they use at the Maple Leaf Foods plant to sterilize the equipment there. And you can also use hydrogen peroxide to sterilize medical devices. There's a couple of YouTube videos if you want to see. In our lab, in the microbiology lab, we use a quaternary ammonium product. So this is a, oh, you can't see it here. Let me just move this. So in the lab, we have this disinfectant called Quattro. It's a product from Quebec. This chemical is in a concentrated form, and what you do is you dilute it down to a 1 in 10 or 1 in 100 dilution. I can't remember exactly how much at the moment, but this is what is in the red bottles in the microbiology lab. So everybody's cleaning off your benches with Quattro. And this kind of disinfectant is also used in restaurants. And I have here healthcare facilities, schools, factories, food factories, industrial settings, and institutional settings. So what they do is they remove any source of contamination with something like Quattro.
Also used in
Yeah. And Brooklyn's got,
I was just wondering, because I was on, air quality. It says that the main pollutant in Oshawa is the-
ozone.
that the-
Oh, it could be from car exhaust. And where do we get car exhaust? Toronto. You can think the wind comes this way from Toronto. Nitrogen dioxide, ozone, probably we're getting a lot of their vehicle emissions this way. This is one of the pollute. I think it's mostly vehicle emissions or factory emissions. There's a spot in Oshawa and in other places on the east side of Oshawa, Harmony and Taunton, where you can actually see the CN Tower. So some mornings when you're out, when I'm driving to work or I see Toronto, you can see, if it's a clear day, you can see all the way to Toronto. But if it's a hazy day, there's sometimes like a smog over the city. And that colored smog, you don't notice it when you're in it, but it does kind of.
Fishing.
Yeah, it's Toronto's in its own kind of.
We look towards Toronto from my place when we get farther up. It honestly looks like a giant cloud in the sky in that direction.
And
see
every,
the snow.
the homes are emitting their gas from furnaces, the cars are emitting gas, factories emitting. So you can think there's lots of different sources of pollution. And it's a clean city. It's just, it's a source of pollution.
I think we're looking at ways to also
Is the idea of electric vehicles, right? The only thing is, we're, anyway, getting off. Okay, so other chemical treatments, chlorine. Bleach is a chemical treatment. So your standard household bleach is about 5% chemical compound, 5% sodium hypochlorite. And so people will dilute bleach about 1 in 10 just to clean surfaces. With something like bleach, you can put it on your surface, but the contact time is needed to kill the organisms. So if you just set down bleach, like a light bleach solution, it'll clean off most of the organisms, but it needs 20 minutes to kill off everything. So you can just put a moist layer down and let it dry on its own. This is why sometimes I say in lab, if you disinfect the surface, it's okay to let it kind of be a little bit wet, but give it some contact time, let it kill the organism. Bleach can kill organisms immediately, but it needs 20 minutes for sterilization. So it is a matter of time. And it doesn't kill all spores, but it can kill some. One thing about bleach is it degrades over time. So if you expose your bleach to air, you should make a fresh solution. Temperature is another way of preserving food. And we talked about refrigeration. Salt. Salt is a way to preserve food. It's an ancient way of preserving fish or meat. I have here, it's probably the oldest known antimicrobial agent. Most microbes don't tolerate high salt. And you see in this image, I've got salted cod from Lisbon, it says. And on the other picture, I have jamon, Spanish ham. And there's this lovely YouTube video, if I had more time to show you. The people, I don't know if it's Spain or Italy, where they're making cured meat. They have these great big massive plants where they store the meat in a cold, salted, they salt it, then they store it cold and let it cure for months. Nitrates and nitrites. Here are two other chemical compounds. They inhibit bacteria, really important to inhibit Clostridium botulinum in cured meats. So they add nitrates. The source of the nitrates is becoming more common as celery salt, celery of different types. And you guys have talked about this because it's in your other course, right? And I have a little breakdown here of how much nitrates and nitrites are in things like hot dogs, bacon, pork tenderloin, ham. Somebody have a question?
No.
Oh. Okay, so next it's organic acids I'm talking about is vinegars. So in something like your dill pickles or your just apple cider vinegar, they have different kinds of acids, whether it's benzoic acid, propionic acid, sorbic acid, acetic acid. I put up two pictures of Canadian products here. I have a picture of Reinhardt's apple cider vinegar and Lakeside baby dill pickles. Anybody ever have Lakeside?
Like you said, I'm just taking note of that too.
What's that?
Oh, we're doing another project on dill pickles.
So there are Canadian sources of dill pickles. I haven't tried any. Has anybody tried any?
Only Paul's.
Only Paul's pickles.
Paul makes pickles.
Oh, you make pickles.
Fantastic pickles.
I make pickles too.
Really?
Oh, yeah.
Really now?
That's all.
Bring them in.
Bring in a jar?
I'm working on my project. I was just looking up what this was.
Yeah, you make cut ones,
What's that?
you make spicy ones.
Well, I add pepper,
Well,
like I call them dragon's tooth peppers. They're like those long red peppers. You throw one of those in. They're kind of spicy. I like them, but that's not for everybody. Some people think it's too spicy, probably.
you know what? Forget those people. Those are not your people. And in life, we find your people. So we are your people. Bring them next week. We'd love to try them.
Yeah.
For sure.
And by we, I mean
1,
me.
2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. It's 12 of you.
And that's probably will be next week too.
So 2 jars probably. Okay. Okay. If you guys would.
I'm his
Distributor.
distributor.
See, but we're paid. I have this horrible problem. I make dill pickles and they don't live.
Pardon?
I make pickles and they're gone within a month.
Oh, we have pickles from 2016 still
No, they
downstairs in the
don't last that
basement.
long in my house.
There's just 2 of us, so if you make,
You might be rolling the dice on those.
I want to try.
Yeah.
it.
Yeah. Okay, so carry on. Organic acid, sulfur dioxide, and sulfites are also compounds used, chemicals used to prevent spoilage. This can be used for dried fruits, wines. One of the most common additives to wine is potassium metabisulfite. This is a compound used to kill off yeast or microbes. Also, it helps preserve the wine. There's two compounds here, nisin, nanomycin. These are preserves produced by microbes. They have sulfur dioxide and sulfites to inhibit molds and yeasts. And here's the questions for this section. Why are highly reactive gases like ethylene oxide, hydrogen peroxide used to sterilize microbes? Question for anybody? Why do we use gases to sterilize microbes? Yeah. It might be cheap, might be easy. Easier than filter. Pardon?
Is the answer because it's more cost effective and easier?
Can be. In the case of ozone, it can be more cost effective than micro membrane filtration. And one of the concerns is it practical to use if they're so reactive? That's based on a case-by-case kind of way, judgment. Liquids such as quaternary ammonium, hydrogen peroxide, peracetic acid, and bleach require a specific contact time. What do I mean by contact time?
It has to sit on the surface for a certain amount of time in order to be accurate, effective.
Yeah, so I was saying with bleach, it needs to sit on the counter for like 20 minutes to sterilize. It can disinfect like 1 minute or 2 minutes, but sterilize 20 minutes. Salt and nitrates are commonly used to preserve foods. How do they prevent microbial growth? Salt and nitrates.
Creating an environment that's not just.
Nitrate is acidic. Nitrate forms an acidic medium that destroys the pH and prevents the growth.
Organic acids like acetic acid, benzoic acid are found in food. Why are they added to foods? Kind of the same reason they lower the pH, inhibit the bacteria growth. Okay, so that's part 4. Part 5, antibiotics and antifungals. I'll kind of whip through this quickly. There's 2 antimicrobials, non-chemotherapeutic agents. This is too toxic for using inside of the body. So in this case, it's used on your surface. Chemotherapeutic agents. These are antibiotics, and they can be used to control disease inside. What is an antibiotic? So I have a plate picture here. This is what I've done is on one side grown bacteria, and on these 3 lanes, I've drawn a line of yeast all the way up to the bacteria. And you can see that the bacteria has this zone of chemicals that come out and inhibits the yeast from growing. So they've got 3 different kinds of yeast, and it's inhibiting this, the bacteria inhibits. So they produce a compound that can be bactericidal or bacteriostatic or fungicidal or fungistatic, and can kill or inhibit your organisms. So who discovered these things? We have two labs to thank. One lab from England is Alexander Fleming. He came up with penicillin from fungi. I'll have a story about him coming up. And another person, Solomon Waxman from New Jersey. He's a researcher who came up with the cure for tuberculosis. We'll talk about him, his work as well. Did he? I don't know about that. I know his lab is in London, downtown, kind of London College. Solomon Waxman was at Rutgers. Antibiotics not effective against viruses. Antivirals are used against viruses. So
I never even came here diving.
many are toxic to humans.
What's that? Which ones?
Antibiotics. If you have too much of them, it can be toxic. Yeah. There's a term called the LD50. This is the lethal dose 50. So at what amount do 50% of the population is it lethal to? 50%. So there is an LD50 for some drugs calculated, but not for humans, for rats, of course. You don't calculate LD50 for humans, but we do calculate LD50s for rats. Some antibiotics are used in livestock and it promotes the growth of animals, but they don't know exactly how that works. They just know it works. There's also fungicides. Fungicides. Wait, question? Comment? No, keep going. Okay. Some fungi, there's antifungals as well. And there are compounds against protozoans. They can be toxic to humans. Okay, that's it for class. That's all I've got for class. But I want to talk a little bit about lab. Oh, and we have a test next week, right?
Do we?
You're saying like, you know.
Okay, I should check my notes, but I can share
Yeah,
that with you.
technically, yes.
Technically, yes.
Technically, yes.
So we've done lectures 5, 6, 7, and this week is 8. So the test will be on those 4 lectures. What are the 4 lectures? This one was sterilization, disinfection. Last week, the test won't have very much stuff from last week's lecture. The maple leaf foods, that's more for information. But I do want you to come up with the names for the organisms like E. coli from Walkerton, Listeria misogynes from maple leaf foods, and Clostridium botulinum. So if you could name those 3, that's really good from last week's lecture. Then before that, we talked about Pasteur and Koch. Their 2 labs and Koch's postulates, the S-shaped flask experiment. What did I do before that? I'll have
So it
to,
will be
Germ theory.
that again, online. That's what I was going to do, just an online test.
Yeah.
Yes, we prefer online tests.
Okay,
Then you can kind of get to it when you have time to do.
great.
And this just
Is this the review you're telling us right now, or are you gonna?
I could talk to the test next week for a couple minutes, but it's just to bring your mind that we have a test next week.
Okay. Nope, that's appreciated.
Yeah. What time is it? 54. I was hoping to be done sooner. Sorry, guys. Lab 7 tomorrow morning is going to be the start of a formal lab. There's a template online. The lab takes tomorrow's class as well as a follow-up class. So you'll do some work tomorrow morning, and then you'll do the follow-up the week after
We have a follow-up from last week.
Great. We have a follow-up from last week's lab. Let me just finish this lab 7 first, and we'll talk about it was lab 4. So the first part of lab 7 is you set up a serial dilution. So you have 4 containers, and the 4 containers are diluted. And then from that, you pour a little bit of the diluted material into 2 rows of 8 plates, forming further dilutions. And then you add agar to a bunch of plates in 4-plate manner. And then you also add bacteria or your diluted media on top of the plates in a spread plate manner. So I'll show you what that means in just a second. And then the week after, you count how many bacteria on the plate. And the number of bacteria is just count the number of microbes that you can see. So if here's a pour plate, so you would add some of your stock. See, here's a stock onto a plate, and you can add 1 ml or 0.1 ml, and then you add agar, swirl it around, and then come back a week, see how many microbes there are that have grown in there. The other way is you have a solid plate. I don't know if I will have you pour the media or if you'll just get solid plates. I'll find out tomorrow. You have a plate, you add media on top, and you take a glass rod and you spread it around. So in total, you'll get about 20 plates from the lab tomorrow. Okay. And so it should look like this, where you'll get some plates where there'll be way too many bacteria there to count, too many to count. And then there'll be a plate where you might be able to count in 2 different dilutions. And then you'll make a table like this. This concentration, too numerous. This concentration, too numerous. TNTC, too numerous to count. And then a number where you're actually counting them. And then you can say too few to count. So it's either too numerous, too few to count. If there are just 3 or 4, you go ahead, put the number into the table. And then there'll be a calculation for you to calculate how many there were in the original bottle. Okay, and that's what the lab will be tomorrow. Paul asked about the stuff we did last week. I don't know how many people are going to get results from last week's stuff, because when you poured the media, it cooled down really fast. And when you transferred microbe to microbe, you may have broken up the plate. You may see results, you might not see results. But don't be disappointed if it didn't work out. It's just showing you that you can isolate these things. Okay. Questions? Any other questions online? Any questions, guys? I'll say questions. Amanda, go ahead.
So tomorrow's lab being a formal lab, but our notebooks are stuck in the microbiology lab. How are we supposed
You
to?
bring me the yellow pages. You take out the yellow pages and you use the yellow pages to make up the formal report. And the formal report is online. So you'll just fill in the report. Yeah. And Natalie's got a question. No? Any? Okay. Yeah, thanks guys for listening. We'll see you guys tomorrow morning. Thanks, Robert. See you tomorrow. Signing out. Have a good night.